Hi, my name is Isabella Carmonte, and here's my audience for today. Hi! Because social distancing, one audience member. Alright. Today I am presenting on the long-term persistence of Borrelia burgdorferi antigens and DNA in the tissues of patients with Lyme disease. These are the things I'm going to go over. Do an intro, research objectives, methods, results in discussion, and a conclusion. Okay, so Borrelia burgdorferi is the bacteria that causes Lyme disease. It is usually treated with antibiotics, commonly doxycycline, and doxycycline is very effective against the early stages of Lyme disease when the bacteria is in the spirochete form. But during chronic Lyme disease, bacteria takes the form of biofilm colonies, and it is ineffective against biofilm colonies. Borrelia burgdorferi is a pleomorphic bacteria. It can change in between a bunch of different forms, so it already evades antibiotics pretty well without biofilms because it can go between the rod form and the cockeye form and the spirochete form. So adding on to the mix, being able to form these colonies that are well insulated and antibiotic resistant makes Borrelia burgdorferi very hard to treat. So my paper that I picked focused on this woman who was treated with antibiotics for a very long period of time. She had Lyme disease for at least 16 years, maybe more. She showed symptoms starting in 1987, and she passed in 2003. But she may have had it longer, just been asymptomatic. Before Lyme disease, she was a high-functioning pediatric intensive care nurse, but afterward, she was went through a lot and was no longer able to work. So she experienced regular symptoms of Lyme disease, such as neurologic deterioration, joint pain. She received treatment over a period of over 11 years when she got diagnosed in December 1991 all the way up to 2003. So in May of 2003, her insurance caused issues and she wasn't able to get the antibiotics that she normally got. And then shortly after, while they were trying to fix things with the insurance, she still wasn't getting her antibiotic treatment, and she went through a series of grand mal seizures and passed away from that. So what are biofilm colonies? So that's what we'll be looking at, and that's what we'll be looking for in this woman's autopsy tissues. And biofilm colonies are aggregates of loose planktonic microorganisms. So they are clusters of bacteria insulated with a bunch of extrapolymeric substances that keeps them antibiotic resistant. They're 1,000 times more antibiotic resistant than free bacteria. Antibiotics cannot penetrate the matrix, and if they happen to, the microenvironment inside of the matrix is also antibiotic resistant. So they don't have a good shot. And this picture here shows what biofilm looks like. Cluster of bacteria with this gel-like stuff around it. That's the extrapolymeric substance that keeps it insulated from the bacteria. So it's insulated, like we just talked about, and it's held by this extrapolymeric substance that enhances protection and adhesion. Common biofilm markers are alginate, extracellular DNA, calcium ion channels, and protrusions. We will be focusing on the biofilm marker alginate today in this presentation for confirming the presence of the biofilm colonies. The biofilm colonies are one of the main reasons that chronic disease are able to persist in the presence of antibiotics and are incurable. So here I have a picture of just a regular sodium alginate solution to show you what alginate looks like and how it contributes to that matrix of insulation and protection. So the research objectives that we'll be answering today are: does Borrelia burgdorferi persist in the presence of antibiotics? And So when does it persist in what form, and was it present in this woman's autopsy tissues past the 11 plus years of antibiotic treatment, and if so, was it mainly found in what form was it mainly found in her autopsy tissues, and were there any inflammatory markers present? And by that I mean, are we going to find lymphocytes surrounding these colonies? And that would, so we're looking at that because of the chronic inflammation associated with Lyme disease. So the methods that I'll be going through are autopsy, tissue procurement and processing, and then we'll look at the immunohistochemical staining process that was analyzed with fluorescence microscopy and confocal microscopy, and then fluorescent in situ hybridization was also used as another method. So an autopsy was performed 5 days after she died. Her body was preserved with formalin fixation, and the tissues from the brain, liver, heart, and kidney were embedded in paraffin and stained with hemotoxylin and eosin. So immunohistochemical staining was done on the heart, the kidney, the liver, and the brain using Borrelia burgdorferi and alginate specific antibodies. And these findings were quantified by looking at the number of biofilm colonies in each tissue and the size of the biofilm colonies in each tissue. So we'll look at that later. And the control for this experiment, we used just a woman who was close in age, so a woman who was close in age who did not have Lyme disease and didn't have other pathological changes in those 4 tissues that we're analyzing: the brain, the liver, the kidney, and the heart. And they were also stained with the Borrelia burgdorferi and alginate antibodies. So her tissues were stained with those just to see if these antibodies were truly specific for Borrelia and alginate. So we used, in this experiment, they used, not we, 2 different labeled secondary antibodies for alginate. So some of the results you'll see that alginate is red, and some of it you'll see alginate's blue. So fluorescent red tag was used for the immunohistochemical experiments that were analyzed with fluorescent microscopy, and fluorescent blue tag was used for the immunohistochemical experiments analyzed with confocal microscopy, and also a blue tag was used for the fluorescent in situ hybridization experiments that were analyzed with fluorescent microscopy. So over here, this just shows how secondary antibodies work. Here's your antigen, and in this case we would have our primary antibody, and then our secondary antibody, which would be attached to the label. So this is what would have the fluorescent tag, and so that we could visualize with fluorescent confocal microscopy. So fluorescent in situ hybridization was also used as a method for detecting Borrelia burgdorferi specific 16S ribosomal DNA. So they designed a 32 base long probe to hybridize with it, and how that works is you denature the DNA, and here's the probe coming in, and it's attached to this 6 fluorescent amidite at the 5' end, and when it hybridizes, it glows, fluoresces. So that was used to hybridize, and then also a random 32 base long probe was used as the negative control, and it was also synthesized and labeled with the same 6 fluorescent amidite at the 5' end. So then the last part of this experiment looked into then taking those Borrelia burgdorferi alginate positive tissue sections that were already immunostained and shown positive for Borrelia burgdorferi and alginate. So if there was a confirmed presence of biofilm colony, looking further and saying, okay, is there CD3 positive T lymphocytes surrounding the biofilm colonies. So CD3 T lymphocyte antibodies were used to see if there was inflammation correlated with biofilm colonies. So the primary antibody used was CD3 specific primary antibody, and then like we talked about before with secondary antibodies, the secondary antibody was used here that was bioaccumulated, and then it was visualized with colorimetric stain and counterstained with hematoxylin. Now we're going to the results and discussion. So, immunostaining with a specific Borrelia burgdorferi antibody and visualizing with the fluorescent microscopy showed the 4 tissues to be positive for Borrelia. And so here we have green fluorescent tag on the Borrelia colonies and red fluorescent tag on the alginate here. The presence of alginate in the same vicinity as the Borrelia showed that the clusters of Borrelia burgdorferi were truly biofilm and not just clusters of free spirochetes. So the alginate is a well-established biofilm marker, and the presence of it there showed that those clusters were indeed biofilm colonies. So here is a quantitative analysis of the immunohistochemical experiments on Borrelia biofilms in the 4 tissues. And you see that the liver showed to have the largest number of biofilms per slide, so their range was the highest, and also the size of their biofilms were the largest overall too. Here is just looking at the aggregates now with confocal microscopy versus fluorescent microscopy that we looked at before. The confocal microscopy analysis on these sections showed that these spirochetes clustered were indeed, again, biofilm colonies because of the alginate presence in the same vicinity. Here it is tagged with fluorescent blue tag, just a different color, and yeah. So then fluorescent in situ hybridization was used to show the presence of the 16S ribosomal DNA for Borrelia burgdorferi aggregates in the 4 tissues as well. And it was showed positive in all 4 tissues. We have the brain, and it was tagged with fluorescent green label again. And so we have presence of Borrelia DNA there, and in the heart as well, and kidney and liver. And again, we looked for alginate in the vicinity to confirm that the DNA clusters were indeed representing presence of biofilm. So here we have alginate stained with fluorescent blue tag. And here's just the quantitative analysis of the fluorescent in situ hybridization experiments. Again, the liver showed to have a larger number of biofilms per slide, and also a larger size of biofilms in comparison to the kidney, heart, and brain. So here's our last results for looking at the inflammation response in correlation to biofilm colonies. So the results were that CD3 positive T lymphocytes were spatially correlated to the presence of biofilm colonies. So here we have biofilm colonies in the brain, the heart, kidney, and liver, and you can see that there's a lymphocytic response in the brain, kidney, and liver. There was no response in the heart. The heart tissues did not show any correlation to biofilm distribution, but we see a large response here in the kidney and some response in the liver and as well as the brain. So an interesting find was that the amount of CD3 positive T lymphocytes was not directly proportional to the size of the biofilm. So we see a smaller biofilm colony here in the kidney with a large lymphocytic inflammatory response, and then a larger biofilm here in the liver with a smaller lymphocytic inflammatory response. So that was an interesting find. So to conclude, after long-term antibiotic treatment, Borrelia burgdorferi did persist in all 4 tissues, and it was persistent, and it survived past the 11 years of antibiotic treatment, mainly in the biofilm form. And by testing for the presence of alginate, confirmed that these were truly biofilm colonies and not just clusters that weren't biofilm. So, and then it was also found that the CD3 positive T lymphocytes around the biofilm colonies showed that there was an inflammatory response associated with each biofilm colony. So it is very common for chronic Lyme disease patients to have chronic inflammation, and this result showed that those biofilms persisting past antibiotic treatment did result in inflammation associated with those colonies. So, yeah, thank you.